All right. So again, welcome. This is Paul Skowinski talking. I'm the president of the Wild Ones Central Wisconsin chapter. I see a bunch of familiar names, and I think there's other people joining from other Wild Ones chapters around the state or perhaps beyond Wisconsin. So welcome. This is our first virtual meeting. We did have the ability to pull off our March meeting in person, but we haven't had any meetings since then. So normally we'd be serving pizza and salad right now, but many of you are probably eating your own dinner right now, provided by yourself. So again, welcome. This is Paul Skowinski talking. I'm the president, and we also have 4 other board members. I think at least one of them is on. Paul Doxey is on the call. Paul is our secretary, and I think at least one more of them will probably be joining, but Carrie, our vice president, will probably be joining in. Ben Kolick is our treasurer, and Karen Blaha manages our website. So the plan for our chapter as far as meetings go is we'll probably continue virtual meetings through at least August. You can see a picture there of our March meeting when we were able to meet in person. We'll still do some garden maintenance type things. The Patch Street project, which if you're a member of our chapter, you're probably familiar with. We did plant a bunch of extra plants there this spring, and we'll need to go in and do a little bit of weeding as the summer goes on. And at this point, we're trying to firm up plans for the July 14th meeting, probably on the topic of common backyard birds, some fun facts about birds, I think. And that would be a virtual meeting through Zoom again. And then August is kind of up in the air at this point. We're, of course, like everybody else, waiting to see how things look at that time and whether in-person meetings are a good idea or not. So stay tuned for more info on that. The Patch Street project is looking nice. I went past the garden site a couple of times in the last week, and things are looking really good. Thanks to Jill, you can see on the left, helping to plant some rough blazing stars. We put in is about 300 plants into the site. There's 2 areas that were about 20 by 20 within this larger site that's probably 20 by maybe 250 or so. Most of it is planted with a variety of prairie grasses with some forbs mixed in, and we had 2 areas that were being taken over by crown vetch. So those areas were sprayed by the Stevens Point Forestry Department last fall, and we went in there this spring and replaced those areas with new plants, all by plugs. So you can see some pretty healthy blazing stars there and some butterfly weed there on the left, and many other species as well. There is a little bit of crown vetch coming back from seed, so that's one of the weeding maintenance items that we'll need to address this summer. And you can see my daughter on the right side helping to plant a few extra plants too. We had some extra milkweeds and other things laying around on our plant rack here at home that we raised from seed, and so we threw a few extra things in there just to fill in a couple of holes. And last week I helped out with a garden at the YMCA's camp about 20 minutes east of here in Stevens Point. The Stevens Point Kiwanis Club was interested in helping out with a project that gets kids studying pollinators. And so I was talking to their president, and I offered to help with a design for a garden and connecting him with a wholesale nursery to get lower prices. And I actually helped out there with the drilling of holes and the planting and watering and such as well. So this was tarped for about 3.5 weeks with a really thick vinyl tarp. And the Kiwanis Club paid for all these plants. I helped to put them in, and we put this little paver border in as well. So we're looking forward to seeing that grow, doing what we can to still stay engaged out there and getting more native plants in the ground despite the whole COVID mess that's going on right now. This was an interesting project in that we didn't really solicit a lot of help because it's such a small site that we can't have a lot of people working in close quarters with each other there. So we had 4 people kind of shuffling around to different tasks within the garden and trying to stay 6 feet apart from each other as we went. So that's it for updates at this point. So I'm going to turn it over to Chelsea Weinzinger, who is the Karner Blue Butterfly Recovery Coordinator. She's going to take the majority of the meeting today. She works for the Wisconsin Department of Natural Resources, monitoring Karner Blues and coordinating the volunteer Karner Blue Butterfly monitoring project. So I will turn it over to Chelsea. And again, you can use the chat function to type questions for Chelsea, and we'll take those at the end of her presentation. Then I will move into a short piece about raising monarchs indoors, and we'll take a few extra questions then at the end of that. So we anticipate being done about 7 o'clock tonight. And with that, I'll turn it over to Chelsea. So I'm going to stop sharing my screen, Chelsea, and then you can start sharing yours.
Okay, thanks. Let's see here. Okay, does that look right to everybody?
That looks good.
Awesome. Great. Well, thank you so much, Paul. Again, my name is Chelsea Weinzinger, and I am the Karner Blue Butterfly Recovery Coordinator with the Wisconsin DNR. I'm out of the Wisconsin Rapids office normally, out of my home now. So I'm just going to tell you a little bit about what we're doing for Karner conservation and monitoring. So just a little bit about me. I got my bachelor's degree from UW Green Bay in 2013, and I liked it so much, I stayed and got my master's degree in 2016. And from grad school, I was lucky enough to grab this Karner Recovery Coordinator position in June of 2016, and I've been with the department since then. So I always like to talk a little bit about why it's important to care about butterflies in general. They make really great ecological indicators. They react very quickly to any environmental changes. They're very sensitive to any negative occurrences, so they can sort of provide this early warning system to other species and the habitat as well, if the habitat is sort of going in a bad direction. This might be a good indicator, as well as indicating the level of biodiversity in some areas. Karners have also sort of become this flagship species for Barron's habitat and its associated species. So anything that's going to benefit Karners is going to benefit that associated habitat. So if you look at this map, this is the national range of Karners. So the bright blue is what is estimated to be the current range of the Karner blue butterfly. And the purple color is the historic range. So you can see it used to be pretty widespread, especially out in New York. And then that grayish color is the wild blue lupin range, and that is the Karner's only host plant. So this gives you kind of a good idea where we're at nationally. Again, this map is probably 3 years old, just a general estimate, but Wisconsin is one of the last strongholds for this species. In Wisconsin, we have a high potential range map. So this is a model based on different data that we've collected that predicts the highest likelihood of where you're going to find Karners. So we utilize this when it comes to regulatory things, as well as making decisions on monitoring and restoration work and that sort of thing. The type of habitat that Karners can be found in is typically savannas, prairies, and barrens, but they can also be found in right-of-way corridors as well. Pretty much, if you find the host plant and it's in an open area, you can definitely keep your eyes out for Karners. That habitat in itself is important because it's globally imperiled, as well as supporting a whole host of other species of greatest conservation need. As I mentioned, the only host plant for the Karner blue is the wild lupin. This comes from the pea family, can be found in sandy soils, and it typically blooms from May to June. So we are, it's currently in bloom right now. So I'm sure if you guys have been driving down the roads, you've been seeing it in the ditches. It's got a really pretty bluish, purplish flower. So it's pretty easy to spot when it's in bloom. In order to identify it, it's pretty easy to identify. It's got 7 to 11 leaflets and typically grows in clumps. Also, the flower, again, is pretty easy to identify, especially when it's in bloom. But there is a wrong lupin. And so when we're talking about putting out native species, we really want to make sure that we get the wild lupin and not this Russell lupin. This can actually hybridize with the wild lupin, and Karners cannot eat the hybridized version or the Russell lupin. So it's really important that if you're in Karner range or potentially near Karner range, that we're not planting this type of lupin because it could virtually wipe out that food source for the Karners. This one, you can identify it. It's a little bit different from the wild blue. The leaves are typically more pointed, and it comes in a whole host of other colors. The wild lupin is more the purple bluish and sometimes white, whereas this Russell lupin comes in pinks and reds and yellows, and it's more robust as well. So it's going to be taller and bigger than your average wild lupin. I've seen this sold at farmers markets and, you know, Home Depot and anywhere you go to get like your common garden store plants. So again, just be really mindful if you're looking to put this in, you want to make sure that you're looking at the Latin names so you can tell which one is which. So talking a little bit about Karner biology. Karners overwinter as eggs, and then in the springtime, the larvae emerge. And if you're lucky, you'll get to see ants tending these larvae. So they have this really cool mutualism where the larvae secrete this sugary substance, and the ants will eat that, and then in turn protect the larva from parasites and predators and all that sort of thing. So if you can see in that middle photo, there's quite a few ants on that one Karner larva. They look kind of like little green slugs. And then typically there are, the first flight begins in late May, early June. So the adult butterflies emerge, and then they lay their eggs again and go through the process and have a second flight, which begins early to mid-July. So right now we are in the middle of first flight. It's probably going to be winding down here in the next week or two. So we're still not allowed to go out into the field. So I'm hoping to get out there soon. But right now we've got some other information coming in from other people that are able to go out, so we know a little bit what's going on there. But one more thing about the eggs. So Karners typically lay their eggs singly, and they look like a little donut. So if you can kind of see, it looks like there's a little dimple in the middle of the egg. So that kind of helps you identify an egg, and they typically lay them on the lupin stems of those plants or on nearby vegetation. So how do I identify a Karner compared to other butterflies? So we use these bands of orange arcs on the forewing and the hind wing to tell us that that's a Karner. They also have these blue metallic dots within the arcs on the hind wing. But again, we really want to be focusing in terms of like our volunteers or maybe people that are out there for the first time. We really try to help push that, you know, look for both those bands of orange arcs on the forewing and the hind wing. In order to identify between a male and a female, if you want to look at the top side, and so the top side of the male is really a beautiful, bright, vibrant blue, and surrounded by this black band and white margins on the ends. The female is typically much duller, can be almost brown sometimes, and they also have a band of orange arcs on their hind wing. So again, if you're IDing a Karner, you want to look at the underside. If you're trying to identify by gender, you want to look at the top side. We have quite a few other blue butterflies that you could maybe confuse Karners with. Again, there's maybe one butterfly that looks very similar to Karners. The range does not really overlap, so it's not a major issue in terms of identifying. And that butterfly is called the northern blue butterfly. And I actually studied that butterfly for my master's thesis. And unfortunately, we believe that butterfly is extirpated from the state. So even if it did overlap, there's likely no more in the state. Other species that could be identified, some hair streaks, but again, a lot of other butterflies have tails, or you know, they don't have those bands of orange arcs, so that's really what you want to look for. Now, to give you a little bit of background on the program, the Karner was federally listed as endangered in 1992, and then following that in 1993, the Habitat Conservation Plan was formed. And that is more so the regulatory side of things. So their main goal is to have no net loss of habitat in Wisconsin. Then on the other side, we have the recovery plan, which the main focus of that is to monitor populations and implement projects in order to reach those population goals. So to clarify that a little bit, we have a Karner program within the DNR, and that program sort of has two branches. So there's the recovery program, which is what I'm involved in. We do the monitoring of the populations and focusing on data collection, analysis, that side of things. And then the HCP branch is the regulatory section. So they have an incidental take permit, which will allow for activities to occur while mitigating or avoiding take for Karner. So for example, if, you know, DOT had to put a new road in or do something with a ditch or things like that, they would work with the HCP and try to avoid or mitigate any take or negative impact that might happen on Karners. So back to the recovery program, we have different goals that the Fish and Wildlife Service has set for each of our sections. So we've got two main goals. The large viable population is a population of 6,000 individuals, and a viable population is 3,000 individuals. And so the requirements are different in different areas within the state, but all of the states that are included within the national recovery program have these types of goals. So in Wisconsin, we've broken it down into 5 recovery units. And then within each of these recovery units, we focus on meeting our population goals. So again, monitoring populations is a really big part of this program, but we also do a lot in terms of habitat management. We're monitoring and maintaining current habitat, as well as creating new habitat for Karners as, you know, time and funding allows. We also focus on coordinating group efforts, so things like partnering for grant applications or getting groups together to do on-the-ground work are really important focuses to support this program. We also do a lot of public outreach, so things like today where we're able to educate and get more people involved and talk more about the program is also a really important part of this. So talking a little bit about monitoring populations, we do this using 2 main methods. So first method that we use is just our presence absence surveys, and that's just how it sounds. You know, we're going out to different areas to determine if Karners are present or not. And that is really heavily focused within our volunteer program. So we really heavily rely on our volunteers to help collect those types of data. The second type of method is called distance sampling, and this is a much more intensive and expensive way to collect data. So we usually contract this work out. And this is a really good way to calculate abundance. So we're actually able to go to an area and get an estimate of the population size at that specific area. We use this type of method for very large populations of Karners. So we use these methods on a handful of public properties as well as private properties. So we have 3 long-term volunteer properties where we do distance monitoring, which is awesome because again, this is a very, it's a more intensive method. So getting volunteers involved to do this is important, especially since they have those higher abundance populations on their properties. So we're able to continue to gather those data and use that valuable information to contribute towards our goals. So this map just gives you an idea of what 2019 looked like for the surveys that we conducted. The red dots are from our volunteers in the field. These are all the different sites that they were able to go to. The blue dots are our distance sampling method, so that's our more intensive survey. And those green dots are just from a previous grant, so those were very similar to our volunteer surveys. So using all the data that we collect, we're able to run some analyses, and we go to 12 to 16 distance sampling sites, and all of those sites are different acreage sizes. So one way to standardize our data is to look at the number of Karners per acre. And so we've got data going back to 2008. So we're able to get a good idea of what the population has looked like within the sites that we've surveyed. Now, this is not a projection of the entire state. This is just looking at the areas that we've surveyed because that's the data that we have. So if you look at this, it almost looks like we're seeing a cyclical trend of the populations building and then bottoming out and then building back up again, which is can be common for certain insect species. So, and you'll see in 2016, we had a record high of Karners at almost every site in the state. And that's when I was hired. And so it was really exciting time to get into the Karner program, but it's all been downhill from there. So I'm hoping to see some good numbers this year. I'm hopeful we're going to be able to get out and do our counts in July and get some more data. Now looking at this graph, this looks at the population estimates standardized again, but broken down by recovery unit. So if you remember the map that I showed you previously, there were 5 recovery units, but there's only 4 on this page because the other recovery unit doesn't have large enough populations for us to conduct distance sampling. So looking at the data that we do have, you can see that each bar is 1 year and it's the population estimate standardized. And the numbers within the bar are the number of sites that we've surveyed, just to give you an idea. So looking at this, you can see again where it looks like populations build and then start to bottom out and build back up again. And the Moreno Sands recovery unit seems to be one of the most stable recovery units. That is the area where we have the most sites. But again, you know, we've got a lot of volunteers in that area that want to have their data counted. So that could be part of it. But it's good to just see that in comparison with the other areas as well. So have we met the recovery goals that Fish and Wildlife Service put forward? No, but we are working hard towards it, and there are a lot of areas that have met those goals, not necessarily within the department, but places like Necedah Wildlife Refuge and Fort McCoy have excellent Karner populations. and they meet their goals each year. And places like Sandhill Wildlife Area, which is a DNR property that continually meets its goal. And again, I mentioned that Moreno Sands Recovery Unit. So there are areas that are doing very well, but those recovery goals were written in 2003. So Fish and Wildlife Service plans to reassess the goals. And so things might be changing in a few years, but right now we're just going to keep moving forward and trying to continue to benefit them in every way that we can. So how do we do that? How do we improve populations? Well, we can't control the weather as much as we would like to. So that's out. You know that insects are very weather dependent and sensitive to those types of things. So then we think, all right, what can we do? Well, habitat naturally is a thing that Karners are fighting against because they're an early successional species. So, they're just going to naturally be fighting that natural succession. So one way that we can benefit Karners is to manage habitat. And we do that by utilizing a lot of different methods. So burning, mowing, herbicide application, all kinds of stuff that can benefit those areas. Now, we're not just going out and burning it as hot as we can, as big as we can. We follow the recommendations that the HCP has, because there are protocols in there that we need to follow to make sure that we're benefiting the species, we're not destroying the species. So there are ways that we can implement this management to benefit the species. So if you look at that top picture of the burn, that was a burn that we conducted 2 years ago in Eau Claire County. And we saw an excellent response from lupin and Karners after. And then the two bottom photos is a before and after of some clearing that was done, I want to say in Wapaka County, maybe. But you can see it took a lot of the scrub oak out of there and opened it up, doing a lot of good. So how can you guys help? How can you get involved? I am always interested in talking more about the volunteer program. So that is a really great way to collect data. You can do it on your property, on public property, wherever you're able to go. We can give you guidance on, hotspots that we're trying to shoot for, or you can just go out and maybe you're hiking one day on a trail and you see a Karner, and you can still submit that incidental observation. We make it really easy to submit your data. You just need your smartphone. So we use the iNaturalist app. So if any of you are familiar with that, it's a really user-friendly app. It makes it really easy to submit data. Another way you can help is to recruit more volunteers. So if you know friends or family that might be interested in something like this, I encourage you to reach out to them and share my contact information, or on the next slide, I'll share the website that you can go to check out the volunteer program. And another way, again, as we talked about before, planting that wild lupin and other native forbs, which is right up your alley. So, that's a really important thing, and all pollinators will benefit from that sort of thing. So talking a little bit about the volunteer program, we started this a few years ago. So this is the 3rd year that we have been doing it. And with COVID, we have struggled a little bit this year. We have not been able to have any of our trainings. So we have not been able to train any new volunteers. We're hopeful for old or pre-trained volunteers to go back and maybe check out some areas, when it is safe to do so. But our main focuses within the program are to gather quality data. So we're looking for lupin data, which we use to help update that high potential range map. We're looking for those butterfly data, the presence absence. That's going to give us some occupancy estimates, as well as colonization and extinction rates. So that's going to help us with our recovery knowledge, as well as updating our range map. And both of those data aid in any type of habitat management. So the more information we have, the better decisions we can make. We also focus on engaging with the public. You know, a lot of the trainings that we do, we hope to train volunteers, but a lot of people just come because they're interested in Karners or want to learn, you know, what else they can do. Maybe they aren't able to collect data, but maybe they can put in a pollinator garden and they're in the middle of Karner habitat. So just spreading that knowledge and helping people engage with us is definitely another main focus of this program. So if you guys want to jot down that link or take a screenshot, otherwise I can always forward it to Paul and he can forward it on to you. But that website link is the volunteer program website. So you can learn more about the program, as well as all of my contact information is listed on there. So if you have any questions, you can definitely reach out to me directly. So that wraps up my presentation. So I'd be happy to try to answer any questions if you guys have any.
There are a bunch of questions in the chat.
Okay.
First one is, do you know if wild blue lupin will grow in soils other than sandy?
So I do know there were some tests done, and I think it could, but it wasn't great. I could look for the study and send it out, but I don't recall offhand. It's unlikely. You don't get super healthy lupin out of it. I think that's why people like to try to purchase the other species of lupin, because that will grow pretty much anywhere.
Okay, we had a couple of questions about attracting lupin. If you simply plant lupin, would you expect to see Karners show up? One of those questions, I think, she mentioned she lives in Eagle, Wisconsin, which is near Waukesha. What if you plant lupin and you're currently outside of the area where Karners are known to be common? Do you think we could expand the range by planting more lupin?
Yeah, that's a great question. So we know with climate change as well, butterfly ranges shift northward. So if we have Karners coming, you know, from the south up, that's definitely an option. And that's actually a focus of part of our restoration efforts, is we're trying to make sure that there is habitat to the north of current populations. So that's an option, as well as, you know, we don't know where every single Karner population is. And that's because Karners are locally abundant in Wisconsin. So if your best bet is to look at the that map, and you can find that on the Wisconsin DNR website, and find where you are associated with that. And if you're in the blue or near the blue, I would definitely encourage it. I think that you know, you never know. Sometimes the animals can end up in weird places. So I definitely encourage it.
Okay, great. Samantha wonders how the sampling actually happens. You had that the slide on that showed people carrying these long poles. Is that used for the sampling, or what is that for?
Yeah, so that pole is broken up into different colors, and all of the colors represent a distance along that pole, because each person walks a transect. So they're walking a straight line from point A to point B, and then they're holding that pole perpendicular to what they're walking. And then they count the butterflies that cross the pole, and they say, all right, you know, 2 butterflies in blue, 3 butterflies in orange. And then those distances are used to calculate the abundance. I could go in detail, you know, for 2 hours talking about the methodology, but hopefully that sort of sums it up for you.
Okay. And do people actually, are you just observing adult butterflies then flying?
Yep.
Okay. Okay. Lisa wondered if there's information about other recovery efforts in other states, particularly Indiana. Are there any efforts going on, and how are things going there?
I don't know offhand. I'm sure your best bet would be to go to the natural resource area within each state. I know that the population there, they believe, is extirpated, so they don't see Karners in Indiana, as far as I know. And I can't remember. We have a federal call where all of the states, work together to talk about what sort of restoration efforts they're doing. And I don't recall offhand what Indiana is doing, just because they're not seeing Karners there anymore. So it's a good question, but I don't know offhand.
Okay. And are you planning on still doing in-person countings this June and July?
So typically we do our counts during second flight, and we do have our contractors set up to do the distance sampling on the second flight in July. So we should still be able to gather those data, which are the bulk of what we do our statistics on right now, just because the volunteer program was still relatively new. And then once the DNR is allowed to go back into the field, that's when we will encourage our volunteer program to also go back into the field and try to do any counts that they can.
Okay. What kind of herbicide do you use to control things?
That's a good question. So I'm not the one that's on the ground spraying the herbicide. A lot of crews do that. But there is an herbicide guideline within the Karner HCP that the crews are required to follow. So I don't know offhand, but that is something, again, that is accessible on the DNR website. I'm just not in the HCP world, so I don't know all of those protocols offhand.
Okay. Susan has a question that I also have. Will growing wild lupin in just a small backyard habitat make a difference, or do they require larger habitat areas?
No, you can absolutely grow them in the backyard. They are found just in small patchy areas, in small patches along right-of-ways. They don't need a big open area. They're pretty hardy. Once they can get established, they're pretty tough plants. So yeah, I would definitely try it.
I think she was thinking about the Karners. Will she find Karners in a small plot like that, or do Karners look for these wide open areas of multiple
Oh,
acres of lupin?
yeah. You'll sometimes see male Karners flying to do like nectaring. So they'll fly far away from lupin patches or in between lupin patches in search of nectar. But typically females stay closer to their natal patch, which is pretty a large space. If you have one clump of lupin, that's probably not enough. You would probably need, maybe 2 dozen or so solid clumps within a larger area. But you might see one if you live close to an area that has Karners. You definitely might see one coming to nectar, or even check if there's any females at your lupin patch.
Okay. I have about 45 in my yard here. I'm in the city and I've only got a third of an acre, and I think the closest Karner spot is only about a mile away.
Okay.
So I've always wondered if I'm likely to see them show up or not.
Yeah, that could be. That could be. You never know.
I hope so. I'll keep watching.
Yeah, sounds good.
Pete wonders if he has 6 acres of prairie with a lot of lupin. I've seen it. It's beautiful. But he hasn't seen any Karners there. And do you think it's because he hasn't noticed the Karners, or are they just not liking his prairie for some reason?
That's a good question. I don't know offhand. I'm not sure where he's located. It all depends on your surrounding area. So Karners aren't like monarchs where they can travel large distances. And again, the adult Karner only lives 7 to 10 days. So they aren't out for very long, and they aren't traveling large areas. So in our protocols, we estimate conservatively that for a Karner to establish in a new lupin patch, the lupin patch should be 300 meters away maximum. to reestablish. So again, that's conservative. There have been papers that have said Karners can travel, like a kilometer or two, but conservatively, we say 300 meters in terms of regulatory. So he might be in an area where he's too far from Karners and, he's boxed off. Or maybe there are Karners out there and, it's difficult to see them sometimes, if there's a lot going on or that sort of thing. But hopefully now, with the identification that we went through, you can keep an eye out for them and maybe they'll show up.
Okay, we had a couple of related questions come in, wondering if it's possible to transplant or import Karner blue in some stage or another from one place to another to encourage population growth in a new area.
Yeah, so that definitely is a possibility, but we would need a permit from Fish and Wildlife Service. So it's not something that we're able to do just on our own, like even the DNR, because it is a federally listed species. We would need to make sure that we had all the legal paperwork done to allow us to do that.
Okay. Do you know about efforts in Minnesota? Anything you can tell us about that?
I do know that there was habitat restoration work done just between so up in Burnett County in Wisconsin, right across the border. There was some work done, I believe, on St. Croix State Park, I want to say. And so, they're continually doing work there and waiting to see if Karners show up.
Okay. Richard asked if there is any introgression in the hybridization of the two lupin species.
That I do not know. I do not know anything about specific genetics, so that would not be a question I could answer.
Okay. All right. I think the last question is Cindy's astonished at the short life cycle or the short adult life stage of the Karner blue, and she's wondering how many eggs they lay during that 7 days and how many eggs will actually see adulthood.
Ooh, that is an excellent question that I don't know the answer to, but I am going to look up because that would be great to know. I do know that during first flight, Karners will lay only one egg at a time per stem that they're on. And then in second flight, they will lay maybe 1 or 2 eggs per lupin stem. Because the thought is over the winter, it's unlikely for both eggs to survive. So they're wanting to kind of bulk up their chances, I guess, over winter. But I don't know about the egg survival rate. I'm going to see if there's any papers about that. And if I find anything out, I'll email you.
Okay. I have one more question about eggs. I think we got through all the ones that are in the chat. You had, I think, mentioned that the eggs overwinter.
Mm-hmm.
So there's a couple of egg laying periods during the summer, and then the ones that are laid in August or the
Mm-hmm.
end of the second flight period, then are there all fall and winter and spring?
Exactly. Yep. So they're there for months. And
Okay.
so that's why it's super important, wanting to have that snow cover for protection.
And those eggs are laid on stems that stay there
Yep.
for the winter?
Yep.
Okay. So it would be important then to keep the stems of the lupin up until spring so they can hatch.
Yeah, definitely. You wouldn't want to clear out any of the lupin or debris. You definitely want to leave that there. And then the new lupin would shoot up, and the larva, once the larva hatch, they find that new lupin. So even if they're in the leaf litter at the bottom, they'll still find that new lupin. And then once they found that, they're a little bit safer. But yeah, that's definitely important to leave that.
Okay. I think a lot of our members have lupin growing and would be interested in knowing the proper timing then to cut them back. If they were going to cut back vegetation in the springtime, should they just wait until the lupin has emerged and is 6 inches tall, and then it's safe to assume that the eggs have hatched and they've moved on to the new lupin?
Yeah,
Or,
yeah, and it's a little bit difficult. I mean, obviously, it all depends, with your conditions that year, right? If it's, rainy or wet or dry. But I would say I would I would estimate that once lupin blooms, you're probably fine. Just because I'm gonna be more conservative about that. If we do have Karners in that area, they're likely going to be emerging when the lupin is blooming. So you're probably safe to do it at that time.
okay. And one last question came in from Pete, wondering how burning affects the egg survival.
Yeah, so when it comes to burning, as I mentioned, we have a specific protocol that we follow, and we only burn a specific portion of an area. And when we burn, we know where our occupied lupin is. So that means that the lupin is occupied with Karners. So I'm not going to burn all of my lupin, because if that's the only lupin that is occupied, there's no to a low chance of that area becoming occupied again. So we want to make sure that we're saving some portion of occupied lupin for those Karnas to be able to reestablish that new burned area. So again, in terms of large scale burning, some of those some of those eggs might not survive, but we are reserving, a specific area. So I guess it's really dependent on how big of an area you're burning, and what your goals are, but there definitely is guidance to help you with that.
Okay. And Pete was also wondering if he should not burn to encourage blooming of the lupin.
So, well, if you're just focused on the lupin, it responds very well to a burn and disturbance in general. So if you're not in an area that is high potential for Karners and you're just focused on the lupin, then yeah, that will definitely help your blooming. But if you're looking at, okay, I want to be more conscious of Karners, the first step would be to conduct some surveys to see if you do have occupied lupin in your area. And then if you don't, then you can burn, and you can burn it all, really. You could totally just take out that area and then say, okay, well, I didn't find any Karners, so I'm just going to go ahead and redo this entire area and maybe encourage, way more lupin to come up. And then, you wait a year or two to do any other type of burning. So that's another, I guess, protocol that is in place, is the timing. So we're not going to burn an area again and again. We're going to wait a few years in between. So Karners respond really well to burns between 5 and 7 years apart.
Okay. And Samantha asked what species of ant tends the eggs. Richard chimed in and said Wikipedia lists several species. Do you have anything you want to mention on that?
I don't. I haven't done a study on ants, and I think it would be super interesting, but I don't know offhand what species.
Okay. And are they tended because they excrete something like aphids do, that the ants are getting something from it, or why are the ants interested in protecting the caterpillars
Yep, that's exactly right.
or
So
the
the
eggs?
larva secrete that sugary substance, and then the ants eat that. And so they're always tending those larvae, taking those secretions off and keeping them clean and protecting them from, parasites and predators.
Okay. What does the caterpillar look like?
It kind of looks like a little green slug. It's like a lime green, and it's got a darker green strip going down the middle of its back. It's really hard to tell where its head is. It doesn't have any distinct markings like that or anything. But it, again, it just kind of looks like a little green slug. It's cuter than a slug, but still slug-like.
Okay. All right, that's all the questions from the chat. Were you planning on sticking around until the end, or if there are other
Yeah,
questions that pop up?
I can stick around, yeah, in case there's a few more questions.
Okay. All right, so we'll take questions after my piece on monarch rearing. If people have questions for me or Chelsea, we can take some more at the end there.
Sounds good.
All
Thanks.
right, thank you very much, Chelsea. Okay, so then we'll move into monarch rearing indoors. This is something that a lot of people have have been starting to do in recent years to respond to the perils of the monarch population in Eastern North America. And there are definitely some ways to do it correctly and to do it incorrectly, where you may be causing far more harm to the monarch population than good. And so I wanted to just run through a few of these things to consider if you're thinking about raising some of these inside. These are pictures from my house here. The left one is our backyard, and that's my daughter, who really enjoys raising butterflies, and we do have a few in the house already here. And she just loves counting them every morning and writing down how many are in each life stage as the first thing that she does every day. So it certainly can be very enjoyable as a family activity. If you're gardening for monarchs, keep in mind that there are 12 species of milkweed that are native here in Wisconsin. Not all of those are easy to obtain or easy to grow, but the important thing to remember is that there's more than just common milkweed. So for someone who doesn't want to plant common milkweed because sometimes it can be aggressive in small areas, there are other options out there, and the monarchs will use those just as well. So butterfly milkweed or swamp milkweed or poke milkweed or showy milkweed, those are all options that you could choose as alternatives. The monarch adults need more than more than milkweed. They will feed on the nectar of milkweeds, but they need other things that bloom throughout the season to keep them fueled throughout the whole season instead of just when the milkweeds are blooming. So keep that in mind. If you are trying to plant a monarch garden, you need more than just milkweed in there to really sustain monarchs and keep them around. Late season flowers, like many things in the aster family, goldenrods and different asters, boneset, Joe Pyeweed, things like that, those are all great ones for fueling monarch migration. They bloom late, and the monarchs can fuel up on that nectar before they leave to migrate to Mexico. And if you're raising any of these monarchs indoors, you have to do it in a clean and safe environment for them. If you're going to put them into a dirty enclosure and it's not cleaned regularly, or they're not supplied with a fresh supply of food regularly, then you're just going to be raising a bunch of unhealthy, suffering monarchs, and that's not really what we're aiming to do. So you can bring them in different life stages. The safest time to bring them in is probably in the egg stage. This is a monarch egg that we brought in last year. We actually have a video of this egg hatching, which was kind of cool. So when you bring them in as an egg, they have less chance of them being parasitized. The caterpillars sometimes get parasitized by other insects, especially the tachinid flies that lay eggs inside the caterpillar. So you're less likely to find out you have a parasitized monarch if you bring them in very early, before those parasite insects have found them. So once they hatch out, you see a caterpillar that most of you are probably familiar with. They take about 2 weeks in the caterpillar stage, and then another 2 weeks or so in the chrysalis stage. So this is a caterpillar more than halfway through its life stage. And when the caterpillar is ready to turn into a chrysalis, to go into the pupa stage, it spins a little web of silk and then turns around, and it attaches some hooks at its back end into this web, and then it hangs down in a J pattern and eventually molts its final time and turns into the chrysalis stage. And you can see that web up on the top there. And they spin around and they really weave those hooks in and get them really tangled up in that silk, so they can lay there undisturbed for a couple of weeks and be resistant to wind and things like that if they're wobbling around. So then you end up with this chrysalis here, and it's this nice green color with these gold flecks in there for the first week and a half to nearly 2 weeks. And then the last day or so of that pupa stage, when it's in the chrysalis, it starts to turn this transparent or turn transparent on the outside of the case, and you can start to see the actual butterfly inside and all the colors on that adult butterfly. And then they'll hatch out, and they'll be a little bit crumpled up like that, and with a really fat abdomen for an hour or so, and then they start to pump all that blood into the veins of the wings, and they stretch them out and dry them, and then at that point they can fly. So that takes another hour or so for them to dry out, and then they can fly off. So things to consider. One of the really important things is that if you have a monarch in the chrysalis stage, it should not be allowed to hatch where other caterpillars are feeding underneath. If you just have 1 or 2 caterpillars, then you probably don't have to worry about this too much because they'll be in the chrysalis stage at the same time. But if they have a parasite called OE, which I'll talk about a little bit in a minute here, the spores of this protozoan parasite are released by the adult butterfly as it hatches out of the chrysalis, and then for periods after that as well. But you don't want them to open up from their chrysalis and then scatter all these spores underneath them where other caterpillars are feeding, because the parasite spreads by the caterpillars ingesting the spores of the parasite. They get inside that caterpillar's body, and then they do damage to the caterpillar and move to the outside of the butterfly as it goes through that pupa stage. And so that's how they spread, and you don't want the spores to be shedding down on milkweed that other caterpillars are then eating. So what I do here is you saw how I have the screen lids on top of the tanks that we use. And as the caterpillar goes up there and it spins its chrysalis with that silk, it's in a metal screen, and so I can use a sharp tweezers to actually pull that webbing off, and then I move that webbing to a dedicated chrysalis tank. And here we have fishing lines strung across from side to side, and then I take a piece of transparent tape and I just sandwich that over the fishing line and onto that webbing, and then it hangs there just as it was hanging on the screen lid of that tank. So they hang in there. I do have a lid on there, so if they hatch out and I'm not there at that moment, they can't actually fly out and into the house. They'll stay in the tub for a little while until we are able to let them go. So this is what they look like when they're hanging in that tub. And we check all of them for OE, the Ophriocistis electroscira. It's usually called OE for good reasons. It's a parasite that affects the monarchs, particularly as an adult, and often results in very crumpled up wings and weak monarchs that can either not fly or sometimes can't even stand up. They'll often just die laying on the ground under their chrysalis because they're too weak to move. Occasionally, though, you do see heavily infected adults that can fly perfectly fine. And that's obviously important to the life cycle of the parasite, because they need some adults to be able to move around and keep shedding the spores of this parasite. But often you'll find these monarchs that they hatch out and they can't even move, and they'll just slowly die because they can't move and certainly can't feed. So we do look at every one of our monarchs that hatch out under a microscope. You need about 100x to 400x magnification to see the OE parasites. And you can see my daughter there is looking through the microscope at a slide. What you do to check for OE is to take a piece of clear tape and you stick it on the abdomen of the butterfly, and that pulls off some scales, and you put that onto a clear slide, and you can look at that under a microscope and see you're looking for these little dark football shaped clusters on the scales of the butterfly. So obviously not something that you can do by sight. You actually do need a microscope to be able to see these. And so it's often discouraged to raise any large quantities of monarchs, because you won't know it if you're just releasing a bunch of infected monarchs out into the wild. You're actually doing more harm than good. So it's something to consider. There are volunteer programs where you can collect these samples of scales and you can send them off to have someone in a lab analyze them and tell you if you have OE on your monarchs or not. So I also would highly recommend that you keep close eyes on your milkweed food source. If you're bringing caterpillars inside and you're feeding them milkweed, you don't want to go out and harvest milkweed that's been treated with any kind of pesticide, because then you're just going to kill your caterpillars. We choose to grow our own milkweed here in pots. We do have a bunch growing around the yard as well in different gardens of 4 different species. and we use those. Sometimes we'll cut the tops off and we'll use those as food. But we also want to have some that go to seed so we can collect the seed and we can start new plants in pots next year. So we don't like to harvest all of our plants that are growing in the yard. And we'll grow usually 100 to 300 potted swamp milkweeds every year for the purpose of feeding monarchs in our tanks. And the other thing you see in the photo there is dill and fennel, because we also bring in a few black swallowtail caterpillars and we'll feed those the potted dill and potted fennel. They'll eat anything in the carrot family, but they really like those two herbs for feeding on. So we grow those in pots as well. So this way I know exactly what those plants have seen. They definitely have not seen any pesticides if they're on my property. I've always got a steady supply of them. And we also rent out what we call the Monarch motels. Or not rent out, I shouldn't say. We just loan out these free kits to the neighborhood families with small kids. They come over and they get an enclosure that's a 3-gallon tank with a screen top, and we give them a couple of potted milkweeds and a caterpillar, and they get to take that home and they get to raise up the butterfly. So when that caterpillar eats all the milkweed in their tank, then they just come by and they go right out to the rack in our backyard here, and they swap out their milkweed pots for new ones. And the ones that they bring back will just sprout again, and we'll use them a second time often during the same season. But they just continually exchange their milkweeds. And so that's another reason why we like to have these potted ones, because it makes it really easy for these kids to come by and exchange out their milkweeds to get more. So that's the way we prefer to do it. And here you can see a couple of examples. There's a swamp milkweed on the right and a fennel plant on the left. And these caterpillars eat a ton, so keep that in mind as well. If you haven't raised a monarch before, raise one first before you raise more than one. They eat an amazing amount of food, and one of those potted milkweeds that you saw in that prior photo will be eaten by a nearly mature caterpillar in a day to a day and a half, one caterpillar. So they eat a lot, and if you don't have the food supply available, then you're going to find yourself in a pickle with a whole bunch of hungry caterpillars and nothing to feed them. We also clean and disinfect the tanks at least once a day when we have a bunch of caterpillars in here, and we're replacing the food every day, if not more than once a day. So here's a picture of my daughter a couple of years ago. Those are two 10-gallon aquariums, and the way we like to do it with these potted milkweeds is to stack them on their sides so that the caterpillars are never crawling around in their own feces or someone else's feces. This way there's less of a chance of bacterial or viral issues popping up with the caterpillars. The OE is certainly a problem, but there are other diseases and other health issues as well. And just like raising any animal in a dirty environment, the caterpillars have that risk as well. So this way, all the fecal pellets from these caterpillars go down to the bottom, and the caterpillars are always up above in a clean, safe location up there. And this is what we did every morning over the summer. You can see there are 9 potted milkweeds in the left aquarium. I think there's actually 10 in the right one, but this is after one day. And there's about 10 caterpillars in that tank, I believe. We typically put 1 plant in there per caterpillar, almost per day. So they eat a lot of milkweed. And that's basically what you see from one day to the next day in that photo. So there's our caterpillars going back into the new tank. And I mentioned this before, raise one first. If you're going to try raising monarchs, please do it. Just do 1 or 2 first instead of trying to bring in a whole bunch. It is a really big responsibility and it'll take a lot of time. We probably spend close to an hour a day cleaning out these tanks. We use a dilute bleach solution to clean them out every morning, and we have to replace all those food plants as well and then transfer the caterpillars over between each tank. So it definitely takes a lot of time. I enjoy it. My daughter loves it. So it's worth the time investment to me. But it is definitely a big responsibility. So keep that in mind before you try to take that on. And last thing I'll mention is a couple of really good websites to visit if you're thinking about rearing monarchs or you just want to learn more about monarchs. Monarch Joint Venture or Project Monarch Health, which is monarchparasites.org. There's also Monarch Watch, which is monarchwatch.org. Those are all great sites that have all kinds of information about monarchs and their migration and their biology and rearing tips and things like that. Yeah, one other thing I wanted to mention is that when we raise them inside, we move the tanks out into a 3-season room in August because it's thought that the low temperatures at night and the daily temperature fluctuation actually helps the emerging monarchs to receive the cues to migrate. And if they're in a climate controlled indoor environment all the time, then they don't get that cue that they need to say, hey, you need to start thinking about going south. So we put them outside so that they are exposed to those low temperatures in August and September, and they are able to get those cues that they need to start thinking about migrating down to Mexico for the winter. So that is it for my piece. Thanks again to Chelsea. If there are questions, I'll look in the chat now. And again, we can have questions for me or for Chelsea here, and we'll just keep taking those as long as they roll in. Nancy asked if they are in my home with AC. Yes, we do run the AC. We usually keep it in that area. It's only about 72, 73 in the house, but yes, we do have them in the home with AC on. Charles asked, do you find tussock sedge cats on your milkweed? I think he's talking about tussock moths. Occasionally we've seen tussock moth caterpillars on the milkweed. We do not see a lot of them here. Yeah, I've seen them in the wild more than I have on my own property here. Samantha asked, how often are you finding OE and what do you do when you find it? Last year, I believe we only had 4 out of about 160 that we raised that had OE. The ones that we do find with OE, we just freeze them. They are in rough shape already. They're kind of destined not to make it to Mexico if they're migrating. And if you let them out into the wild, then they're just likely to spread that parasite further. I've done a fair amount of butterfly presentations at elementary schools. And so I will take those frozen butterflies with me in little sandwich baggies. And that way the kids get to see them and handle the bag. So I feel like it's okay to freeze them because I'm kind of putting them out of their misery, and I'm also using them for a purpose that benefits monarchs in the long run. So that's what we do here. Is there a problem with collecting caterpillars in one place and releasing the butterflies elsewhere, like a vacation home versus a permanent resident? There was at least one study looking into whether you could take a monarch from one state and drive it halfway across the country and release it and see if it migrates the right direction, and they do. You could take one from, for example, Ohio and drive it to Minnesota at the end of the season, and they seem to know where they're going, even though they hatched in a completely different area of the country. They still somehow find their way to Mexico. So I don't think that would be a problem. Pat says we have a milkweed disease in Milwaukee County. The plant's yellow and are deformed. Can this be used to feed caterpillars? I don't know the answer to that question, but I would say that it's always best to feed them the healthiest looking milkweed that you can find. So if it does not look healthy, the caterpillars may also sense that and try to crawl off and find a healthier stand of milkweed. So I think it would be best just to avoid that milkweed and try to find a healthier population to feed them. Lisa says, other than keeping the habitat clean, are there other ways to prevent disease? I think the really the most important thing is keeping the habitat clean. It gets the majority of the health issues, I think, would be minimized just by keeping the habitats clean, disinfecting them regularly, keeping them in good health with fresh food and things like that. So the same kind of things you do for your pet or for yourself. It's just best to have a clean environment and have healthy foods available to you and that sort of thing. Occasionally people do, they bleach the eggs, I believe. I've not done this before, but I've heard of it before. Trying to remove any kind of disease or parasites, I guess, that are on the eggs when they bring them in. Um, I guess my opinion is that it's maybe going a little bit too far. So that's just my opinion. I think the majority of the health concerns can be addressed just by giving them a clean environment with good food supply. Let's see. I thought touching and moving the caterpillars a lot was bad as well for them. It sounds like you move them daily and they are fine. Um, yeah, I guess I would think that if you have things on your fingers like lotions or sunscreen or any kind of chemicals that might be on your fingers, those could potentially get into the skin of the caterpillars. I don't know that for a fact, but in our case, we wash our hands before we handle them. And so there's nothing there really to get onto the caterpillars, and we certainly haven't noticed any issues. We have had over a 99% success rate as far as adults or caterpillars living through the chrysalis stage and then hatching out as adults and flying off. So it seems like we're doing things okay here. And I think that minimizing any chemicals or anything that might be on your fingers would be the best thing to look out for. All right, I think I got through all the questions there. We can wait a minute if there's any more that come in. And again, if you have any other questions about Karner Blues or Lupin for Chelsea, we can take those also. Nancy asked, what about when they are in a molting state? Do you move them? No, you do not want to move them when they're in the molting state, especially molting into a chrysalis. You want to leave them alone, and you don't want to move the chrysalis until it's been drying for at least a day. That's kind of a vulnerable time for them. So um, yeah, leave them alone anytime they're shedding their skin. They're moving to the next stage of their caterpillar life stage. And certainly when they're moving into a chrysalis, you need to leave them alone, or they could actually kill them if you disturb them too much. Cindy asked, if you hatch monarchs, do they normally come back to your place on their own? We've seen the ones that we release, we've seen them go uh, well over the neighbors' houses, and we never know if we see them again. We have seen some that go up into the air and they come right back down because we have a couple of very large native gardens, and so there's a lot of flowers. And I imagine if they fly up 500 feet and they look down on the neighborhood and all the different lawns that they see, and then they look back to our house, they probably think it looks pretty good. We have tagged monarchs every year. We usually tag 50 of them in August and September for Monarch Watch. We do see them sometimes hang out for a couple of days, but those are migratory monarchs, and so they typically don't hang around too long because they're starting to think about heading south. Lisa asks, how many tanks with approximately 10 caterpillars in each do you manage at one time? Usually it's only one or two. Sometimes it's a little bit more than that, but we have several 10-gallon tanks here, and we also have all those smaller tanks that we loan out to the other kids in the neighborhood with one or two in each. So typically we'll have 2 or 3 tanks running at any given time, with maybe 6 to maybe up to 10 or so in one tank. Cindy says, do they come, she means if they come back after the migration back north. No, because they are migrating down to Mexico and then they come back to the southern US and lay eggs and die. It is several generations after that the monarch population actually makes it back to Wisconsin. So nothing leaving here in September and October will ever make it back to Wisconsin again. They will only make it back to Texas or Louisiana, and that's about it. So yes, we say goodbye to them forever. Yep, that's correct. All right, well, I don't see any more questions in the chat, so thanks again, Chelsea, for sticking around and for your presentation tonight. Um, look for further communication from me about July's meeting. Again, we think it'll be another virtual meeting like this focused on backyard birds, but keep an eye out for more communication and we'll talk to you again in July.
Thank you so much, Paul. I appreciate the opportunity.
Thanks for being here, Chelsea. All right, looks like there are other Wild Ones chapters even outside of Wisconsin joining us, so that was really cool. And yes, Roberta, there is a recording of this webinar. I'll send it out to the national headquarters as well so they can share it. I'm assuming that's where many of you outside Wisconsin heard about this, so we'll get it out there so people know where the recording is. All right, well, everybody have a great rest of the night.
Thanks very much. Bye-bye.
Bye.